So, good afternoon, students.
Good afternoon, sir.
So, how's your classes going? All well?
Yes, sir.
Okay, so students, I hope you remember last week we were talking about the complement pathway and we did the classical pathway, right? So, out of the three pathways, we have already done the first pathway. And if you remember, last time Shakti also explained uh, this pathway, right? So today also, like we have two classes, so we will try to uh, do the second pathway, which is the alternative pathway. Okay, but before we start with the alternative pathway, we'll again try to understand the classical pathway, because see, if you know the classical pathway, if you have understood the classical pathway in a good way, right, only then you will be able to understand the other pathways. Okay, and I'm telling you, other pathways are very simple as compared to the classical pathway. So if you know the classical pathway, that is the toughest out of the three. Okay, so if you have understood the classical pathway, then other two are very easy. Okay, and other thing is, half of the thing, right, from C5B till the formation of MAC, it's a common pathway, right, for all the three pathways. So you just need to know about the first few steps of the other pathways, right? So before we start with this alternative pathway, let's just refresh what we have learned in the complement system in the previous classes by quiz. Okay, so I'm just going to assign the quiz to you, and once you have done the quiz, we'll discuss the answers. Okay, so let me just assign it to you. Okay, so students, I have assigned it. Okay, so maybe in few seconds it would be visible at your end. I think it's visible now, right?
Yes, sir.
Okay, so start doing it and do it fast so that we can discuss the answers and move ahead. Okay.
Excuse me, sir.
Yes, any problem?
Yes, sir. Sir, at my end, the quiz is showing that it's already been submitted.
Oh, how come?
I don't know, sir. I just opened it and it's showing it's already been submitted.
ठीक है, leave it.
Okay, sir.
Okay, students, start submitting now.
So this is Nandini. It's showing the same thing as it showed Nehal. It says that your response has already been submitted.
So who is submitting on your behalf?
I don't know, sir. I just attempted the quiz.
Okay, students, I've got response from half of the students. So others, do it fast. So students, do it fast now. And students, I also want one of the student to explain uh, the formation of MAC from C5B onwards. Okay, so just prepare yourself. Once we have discussed the uh, answers, okay, then I will start with the alternative pathway, and in that I will tell you how we reach to the point of C5B. Okay, so from C5B one of the student has to tell how we reach to the point of MAC. Okay, so you can start preparing if you want, right? Okay, students, so almost all the students have submitted. I think few have not submitted. Okay, so I think we can now discuss the answers. Okay. The good thing is 16. Okay. Okay. Not so good. Okay. Okay. Now coming to the first question, who won the Nobel Prize for his work on complement system? Okay, so the Nobel Prize was won by Jules Baudet. Okay, so he was awarded the Nobel Prize for the work on complement system. Okay, after that, even Paul Ehrlich worked on the complement system. Okay, and he was the one who coined the term complement. Okay, so question 2 was who coined the term complement, right? And the answer is Paul Ehrlich. Okay, so initial observations were done by Jules Baudet, whereas later on more experiments were carried out by Paul Ehrlich. Okay, and he termed the term complement. Okay, so question 1. So one student has given wrong answer. Rest all of you have given the right answer. Question 2. Uh, 3 students have given wrong answers. Like who coined the term complement? Okay. So 3 students have given wrong answer. The correct answer is Paul Ehrlich. Question 3 was which of the following pathway of complement system is antibody dependent? The good thing is, I think all of you have given the right answer and one student has not attempted. So rest those who have given the answer, so they have given the right answer. Okay, so uh, it's the classical pathway which is antibody dependent. Okay. Now if question 4 is if C2 complement protein is cleaved, the bigger fragment will be. So again, 2 students have said that both will have the same size, but the correct answer is C2A. So C2A will be the bigger fragment. Okay, so C2 is the exception where C2A is the bigger fragment and C2B is the smaller fragment. Right, so it's the only exception. Okay. Question 5 was from which point onward there is a common pathway for all the 3 pathways. The correct answer is C5B, and again 3 students have given wrong answer. Okay, correct answer is C5B. Okay, because from C5B onwards the pathway is common. Okay, what is the role of complement system? Now again, 3 students have given the wrong answer. Uh, One of you have mentioned pathogen killing, 2 have mentioned clearance of immune complex, but the correct answer is all, right? Because if you remember in one of the slide, I showed that, complement pathway and then it can do so many functions. It can kill the pathogen, it can help in opsonization, it can help in inflammation, it can help in immune complex clearance, right? So complement system have a lot of role in our body. It's not just to kill a pathogen. Okay, it has a lot of different role. So the correct uh, answer is all. Okay. Question 7 was what is the need for cleavage of complement proteins? Again, 2 students have given the wrong answer. One of them has mentioned reduce the size of the protein, but that is the point. Why to reduce the size of the protein? And another is activate antibody production, right? But the correct answer is expose the active site. Okay, because if you remember, I with the help of this, I explained you the concept, right? Now, if you have to write using this uh, marker, you'll have to open it up, okay, so that this site is exposed from where you can write, okay. So similarly, the complement protein is in this state, okay, and then due to some or the other factor it is broken down, it is cleaved into 2 components. One is the bigger fragment, one is the smaller fragment, and then on the bigger fragment a site is exposed where the next protein can come and bind. Okay, so when it is in this condition, then the next protein can come and bind to this active site. Okay, now when this binding takes place, it again cleaves this protein, right? So what happens? This protein is again cleaved. So the smaller one it goes away, it diffuses away, right? And a new complex is formed, right? So I think you can also do this kind of practice at your home. You can make, using some paper cuttings, you can make proteins, put C1, and when you open it becomes C1A, no, C2A, C2B, right? And using that you can explain this whole pathway, right? So it's a very interesting pathway, very helpful pathway for our body. Okay. Okay, so that was question 7. Need for cleavage of complement protein. It activates, it exposes the active site. Okay. Now, question 8 is which of the following is the smaller fragment, right? So C3A, C2A, C2, C5B, and C8. Okay, so C2A, as I said, it's an exception, it's the bigger one. Okay, C5B is the bigger one. C8, it does not break down, so therefore it is also a bigger fragment. So the smallest is C3A. Okay, and again, two students have given the wrong answer. They have mentioned C2A. Okay, so just because A is there, it doesn't become a smaller fragment, right? There is exception in case of C2. Okay, clear? So I hope you have reviewed your answers. You now know who has done what. Okay, and although I have the list, whole list, how many students have got what score. Okay, okay, students. So with this, I think if you have any question, you can ask, or else uh, we can start. Any question? Okay, so students, let's see a small video of the classical pathway. Okay, let's understand how this pathway works. Okay, so you have actually seen it step by step, right? You know what happens first, then what happens, then another protein, and then ultimately formation of MAC. So you have already imagined it on one dimension. Okay, so in the video you can see it in two dimension, right? Okay, so just let's see the video in a moving way how it is happening. Okay, so let's first see that, and if you have any question related to classical pathway, then you can discuss. Okay.
Activation of the complement system occurs on surfaces, such as the surface of a bacterium as shown here. In this
Okay, so basically this pathway will start when there is some bacterial antigen. Okay, so we need to have some foreign antigen. Okay, and then this complement system will start.
example, the activation of complement is initiated by antibodies that bind to bacterial cell surface antigens.
Okay, so next point, this because the classical pathway is antibody mediated. Okay, so what happens? These antibodies, they go and bind to the antigen on the surface of the pathogen. Okay, so you can see these antibodies have bound to the antigens present on the bacterial surface.
The C1 complement complex, made up of one molecule of C1q, two molecules of C1r, and two molecules of C1s, binds to the aggregated antibody molecules on the surface of the bacterium.
Okay, now as I said, like because of this binding, okay, because of antigen antibody binding, there is a conformation change in the Fc region of the antibody. Okay, and because of this conformational change, this molecule, the C1 molecule, it can come and bind through its globular head to the antibodies. Okay, antibodies also there in circulation, C1 protein is also there in the circulation, but they will not interact. Okay, they will only interact when these antibody have encountered an antigen, and because of this interaction there is a conformation change in this antibody. Is this point clear? Okay, because anyone can ask you, भाई तुम्हारे ब्लड के अंदर C1 प्रोटीन भी घूम रहा है, एंटीबॉडी भी घूम रही है, तो why don't they link? Okay, they will not link because antibody is made in such a way that this C1 protein will not bind to it until and unless there is some conformation change. Okay, so when the shape of antibody changes, the C1 molecule binds to the antibody. Okay, is this point clear till this point? Students? Okay.
Binding of the C1 complex to antibodies results in the activation by cross-proteolysis of the C1R and C1S proteases. Active C1S cleaves and activates the complement protein C4, releasing a small peptide fragment, C4A, which acts as an aflatoxin. The remaining large fragment, C4B, which contains the labile thioester bond, covalently attaches to the surface of the bacterium through formation of ester or amide linkages. The C2 proenzyme binds to C4B and is then cleaved by the activated C1S, releasing a small peptide, C2B. This leads to the generation of a new protease, C4B-C2A, also known as the classical complement pathway C3 convertase.
Okay, students, so you just saw that first C4 was broken down, okay, by C1S, okay, and then this next molecule C2 attached to this, and then again CS cleaved it, right? So this complex is your C3 convertase, which is C4B and 2A. Okay.
The C4B-C2A enzyme binds C3, cleaving it to release another anaphylotoxin peptide, C3A, while C3B fragments, which also contain a labile thioester, bind to the bacterial surface adjacent to the C4B-C2A complex and associate with C4B-C2A to create.
So students, what is this complex? So what is formed now?
Convertase. Convertase, sir.
Which convertase?
C3
C5.
convertase.
Yes, so this is your C5 convertase now. Okay, so C4B, 2A, 3B. So you have this pink one, your C4B. Okay, this is your 2A, and then the next molecule 3B, right? So this forms C5 convertase, and what this will do, it will now cleave C5.
Create the C4B, C2A, C3B protease that is a C3-C5 convertase. Many molecules of C3 can be cleaved by the C3-C5 convertase.
So you remember I told that at this point there is amplification of this pathway, right? Because by this time the immune system knows that we have to start this system because there is an attack. So at this point there is amplification of this system, right?
Releasing many molecules of C3A. Many of the resulting C3b fragments are able to covalently attach to the bacterial surface, decorating it with many molecules of C3b that can induce phagocytosis of the bacterium. The C3-C5 convertase also cleaves and activates C5. Again, the small peptide fragment released by this cleavage, C5a, is a potent anaphylatoxin and is also the most important chemoattractant derived from the complement system. The large fragment, C5b, acts to initiate the formation of the membrane attack complex, the terminal stages of the complement cascade. In this process, C5b.
Okay, so you can see C5b is formed, and then the next molecules will start coming like the C6, C7, okay?
Assembles with C6 and C7. C5b itself is not membrane associated, and the C7 molecule allows this complex to insert itself into the bacterial cell membrane. C8 also binds to the C5b C6 C7 complex and inserts itself into the cell membrane. The C5b 678 complex catalyzes the assembly of many molecules of C9 to create a cylindrical pore spanning the cell membrane. The pore disrupts the ionic and osmotic balance across the membrane and thus kills the bacterial cell.
So students, do you want to see this again in one go without any break?
Yes.
Is it okay, or do you want to see it again without any stoppage?
Sir, can we view it again?
Okay.
Yes, sir.
So now I won't stop. So just focus. It's a small video. So students, everyone, just focus for 3 minutes on this video, and this whole pathway will be very much clear to you, okay?
Activation of the complement system occurs on surfaces, such as the surface of a bacterium as shown here. In this example, the activation of complement is initiated by antibodies that bind to bacterial cell surface antigens. The C1 complement complex, made up of one molecule of C1q, two molecules of C1r, and two molecules of C1s, binds to the aggregated antibody molecules on the surface of the bacterium. Binding of the C1 complex to antibodies results in the activation by cross-proteolysis of the C1r and C1s proteases. Active C1s cleaves and activates the complement protein C4, releasing a small peptide fragment, C4a, which acts as an anaphylatoxin. The remaining large fragment, C4b, which contains the labile thioester bond, covalently attaches to the surface of the bacterium through formation of ester or amide linkages. The C2 proenzyme binds to C4b and is then cleaved by the activated C1s, releasing a small peptide, C2b. This leads to the generation of a new protease, C4b C2a, also known as the classical complement pathway C3 convertase. The C4b C2a enzyme binds C3, cleaving it to release another anaphylatoxin peptide, C3a, while C3b fragments, which also contain a labile thioester, bind to the bacterial surface adjacent to the C4b C2a complex and associate with C4b C2a to create the C4b C2a C3b protease that is a C3 C5 convertase. Many molecules of C3 can be cleaved by the C3 C5 convertase, releasing many molecules of C3a. Many of the resulting C3b fragments are able to covalently attach to the bacterial surface, decorating it with many molecules of C3b that can induce phagocytosis of the bacterium. The C3 C5 convertase also cleaves and activates C5. Again, the small peptide fragment released by this cleavage, C5a, is a potent anaphylatoxin and is also the most important chemoattractant derived from the complement system. The large fragment, C5b, acts to initiate the formation of the membrane attack complex, the terminal stages of the complement cascade. In this process, C5b assembles with C6 and C7. C5b itself is not membrane associated, and the C7 molecule allows this complex to insert itself into the bacterial cell membrane. C8 also binds to the C5b C6 C7 complex and inserts itself into the cell membrane. The C5b 678 complex catalyzes the assembly of many molecules of C9 to create a cylindrical pore spanning the cell membrane. The pore disrupts the ionic and osmotic balance across the membrane and thus kills the bacterial cell.
Okay, so students, now you have seen the video also, and I'm sure this concept is clear to you. Any questions? I try to make this concept easy so that it becomes easier for you to understand this pathway, right? I hope everyone has understood this, right?
Sir. Sir, can you share the link for both the videos?
Okay, so it's not a link, Yukta. It's like I have downloaded that video. Okay, so maybe I'll find some other video on YouTube and maybe I'll send it to you. Okay.
Okay, sir.
Like I have been teaching this antibody complement, all these things to the bachelor students, and students, even after so many years, they remember all these concepts, right? I just got a message on Teachers Day which said that, sir, the way you used to deliver your lecture was the one I miss the most now, right? So they are missing it even today, and especially the one I liked the most was when you taught the concept of complement system pathways during BSc. Okay, so students, the whole point is that I have tried to make it as simple as possible for all of you, and I just hope that this concept of complement system remains with you throughout your life. Okay, so if you have any question, anything in your mind, you can ask, or else we'll have attendance first and we'll then see how much time is left to start a new topic or not. Okay, so Adish.
Present, sir.
Okay, Atoeba.
Present, sir.
Ayushi.
Present, sir.
Okay, Sam.
Present, sir.
Devanshi.
Present, sir.
Ishita.
Present, sir.
Janvi. Janvi. Kshitij.
Present, sir.
Manaswini.
Present, sir.
Okay, Namrita.
Present, sir.
Nandini.
Present, sir.
Nehal.
Present, sir.
Okay, Rupal.
Present, sir.
Sahiba.
Present, sir.
Sanjana.
Present, sir.
Shakti.
Present, sir.
Shivani.
Present, sir.
Tanvi.
Present, sir.
Tridev,
Present, sir.
Vivek, okay, Janvi, I can see you now. Okay, Vivek, Yukta,
Present, sir.
okay, and Yuvraj,
Present, sir.
Yuvraj, you are not
मैं
visible
जाता
here.
था छोड़ दिया मैंने छोड़
Yes, Vivek. Okay, Yuvraj, Vivek.
Sir, I'm present.
Okay, Vivek, fine. Okay, students, so I'm not going to start with the alternative pathway in this class because just after 5-10 minutes we have another class. But uh, when we were talking about the classical pathway, right? So sometimes there can be some problems also. Okay. Now what happens? I hope you remember what happens. So C5b, then comes 6, and then come 7. And at that point, the complex can get inserted into the membrane of the cells. Okay. Now you just imagine that this thing is going on at a bigger level. A lot of C5b6 7 complexes are formed, right? And sometimes these complexes can get inserted into the membrane of nearby cells. Okay, and they may mediate innocent bystander lysis. Okay, so I hope you know that कई बार क्या होता है कि दो लोगों का झगड़ा चल रहा है, ठीक है, बट मारा कोई और जाता है जो bystander खड़ा होकर देख रहा होता है, right? You know this happens. So it's the same thing. Is sometime what happens when this C5b6 7 complex is formed, it may go and get inserted into the nearby cells. Okay, so जो innocent nearby cells say they can also die. Okay, and autoimmune disease in which immune complexes are formed, tissue damage result from such innocent bystander lysis. Okay, so sometimes this complement, the classical complement activation, this can also lead to some kind of disease. Okay, but again that's a very rare thing. Okay, but yes, this can happen. Okay, and okay, but still, as I said, it is very rare, because this whole pathway, the classical, not only the classical, the whole complement pathways, they are highly regulated. Okay, they act only when required. Okay, so it's not that in our body this is the only pathway which is highly regulated. In fact, all of the immune system is very much regulated, right? When there is a need, it gets upregulated. When there is a need, it downregulates itself. Okay, when the pathogen has entered, right, and it just starts doing its work, right, and how they get the signal, how they come to know that we have to start this or we have to stop this. Okay, so there are messengers, and those messengers are called the cytokines. Okay, and will maybe I'll just have a one class on cytokines as well, right? So what these cytokines do, how they act. Okay, so this one cytokine will give a particular signal to start the reaction. Okay, when it senses that now things are under control, we don't need this, then another cytokine will come and bind to this cell, and it will tell now you stop this work. Okay, no need now. So the cell will stop that function. Okay, so this way the immune system is highly regulated, and even this uh, complement pathway is highly regulated. So maybe in one of the class I'll just discuss briefly what are the different proteins which are associated in the regulation of these pathways. Okay, but again, at your level, bachelor's level, I won't get into the details of how these pathways are regulated. Okay, so don't worry about that. So I won't discuss that in detail. Okay, any other thing that you want to know?
Yes, sir.
Yes, what's the question?
Yes, sir. So just now the case you said. So will our immune system come to know that the our complement system it lies some other cell, um, and not the cell which it wanted to lies?
Mm-hmm.
So will our immune system come to know that or not?
Okay, so see again, obviously when a normal cell will die, okay, obviously it will release its content. Okay, so from that the body will get to know that this has happened. Okay, uh, so that is one way, but if the body doesn't get to know, then it causes disease. Okay, so in that case, what you need to do is you need to shut off the complement system. Okay, because if there is some genetic defect or something like that, then the only solution is that you need to shut off the complement system by some drugs. Okay, or by using monoclonal antibodies. Again, we have read monoclonal antibodies. So you can have a monoclonal antibody designed against C1 protein. Okay, so if C1 molecule is blocked, it will not bind to the antibodies and activate the complement pathway. Okay, so maybe our system doesn't get to know, and obviously then it will create some issues, and then you need to see how to control it. And to control it, you will have to shut down the complement pathway. But yes, if you do that, then the person becomes susceptible to various infections. Okay, so then you need to manage the infections. So there are a lot of things. Okay, so we will be reading all those things when we talk about the autoimmune disorders and all those things, right? Okay, so students, I'll end the class here. Okay, so we'll just meet after 5 minutes for the second class. Okay, and we'll start with the alternative pathway in that class.
Thank you, sir.
Okay. Okay.